Hello everyone. Our today's video is about Sopma Tool, assessment of 2D structure of mutated proteins. Sopma is the abbreviation of self-optimized prediction method from alignment. It is a package to make secondary structure predictions of proteins. Whenever you will try to search it on the Google and will write the Sopma online server, it will give you the very first option with the name Sopma secondary structure prediction. Clicking on it will take you to the home page of this online tool where you will be able to see a small or narrow window for writing the sequence name, and then there will be a big window to paste your protein sequence. All the values by default values will be used to perform this task for all the parameters given here, like output width, number of conformational states, window width, and similarity threshold. Then you will copy and paste your protein sequence in this box and then click on submit, which will show you this picture which is showing you the status of your submitted job, that either the job is in queue or is running. But it does not take much time, and within less than 2 minutes you will be able to see your results, as it was not able to for me to show all these results in on one slide, so I have divided the results on two. First of all, you will see the amino acid sequence, and below each amino acid there will be an alphabet with the specific color. These colors are actually showing the different types of secondary structures, but many types of you might be familiar with many types of these secondary structure, like alpha helix, beta turn, random coil, but some are not maybe some are not known to you. So first let me explain you the different types of secondary structure which we may know from the Sopma tool. The first one is the alpha helix. As we know that the sequence of amino acids in a protein that are twisted into a coil or a helix and it is stabilized by the hydrogen bonds is known as alpha helix. Next is 310 helix. This portion have 3 residues per turn with an angle of 120 between consecutive residues. It is more tightly wound, longer and thinner than an alpha helix with same number of residues. Hydrogen bonding occurs between the every 3 amino acids in 310 helix. Next type of secondary structure is pi helix. Here the hydrogen bonding occurs between 5 residues. It is less stable than general alpha helices. The beta bridge is a short fragment that shows beta sheet like a bending pattern. Next is extended bridge. The extended conformation in a beta strand is about 3.5 angstrom per residue, and the beta strands can be extended as much as 35 angstrom in length. The next aspect of secondary structure is beta turn, which is this portion change the direction of peptide bond by nearly 180, allowing peptide chain to fall back on itself. Then there is a region of which is known as bend region. This is an unstructured region which occurs normally between the secondary structures, and proline amino acid is mostly found in these bends. And then there is random coil. These are found at both the N and the C terminus of the protein. Now you are able to see here on the top all these attributes are mentioned and with their symbols written in the brackets, like alpha helix is shown with capital H and small h, 310 helix is shown with capital G and small g, and similarly pi helix is shown with capital I and small I. So let us see in this picture of our protein that each in the initially the amino acids below the amino acids small h is written, which is showing the helix, alpha helix portion, and it is representing that all the amino acids which are present at the till 12 the amino acids till 12th position are forming actually the alpha helix, which is followed by 4 C's, and C is showing the random coil. It means that the next 4 amino acids are a part of random coil, and so on. And then you can see here at the top the alpha helix. After alpha helix there is written 380, which is actually showing the number of amino acids of this protein which are found in the alpha helix, and which is constituting about 50.80% of the total proteins. And then 310, there is zero value, like zero number of amino acids are shown here for 310 helix, pi helix, beta turn, bend region, ambiguous states, and other states. While for extended strand, we can see that 77 amino acids are a part of extended strand, which are forming 10.29% of the total protein. And in case of random coil, 291 amino acids are shown, which is making 38.90% of the total protein. And below this information you are able to see the two pictures. The picture which is given below is the graphical presentation of the abundance of these different aspects of secondary structures in your protein of interest. Here you can see the peaks of different colors. The blue color is representing the helix, the red color is representing the sheet, and green color is representing the turn, and the purple color is representing the coil, while on the x-axis the number of amino acids are mentioned. And in order to express this relative abundance of amino acids in the different regions of secondary structure in a more simplified way, or you may say in a more easy to visualize way, a diagram which is present above this peak diagram is given, in which you can see the three colors, the blue, red, and purple, which are the showing the alpha helix, the extended strand, and the alpha helix, the extended strands, and the random coil, and so on. The portions which are most abundantly found in your protein of interest. Now the question is, how can we represent this data in the manuscript? Definitely the pictures, you can copy and paste these pictures in the supplementary data, but I think the more proper way to represent the secondary structure of your protein of interest is the tabulated one. This is the table from one of my published papers, and here you can see there are different columns in which the different attributes of secondary structures which we are going to predict from the Sopma tool are mentioned. In the first column here the probiotics are written, but you may write your number of cases, and in the second column the alpha helix and extended strand next, and in next column beta turn, and in next column random coil is mentioned. And now you can write the values of alpha helix, extended strand, and so on of these parts in these columns. And while in place of probiotics you may mention in the first row your normal protein, and then in the successive rows the case 1, case 2, and case 3. And hence by looking at this table you would be able to compare the proportion of the alpha helix in your normal protein as well as in the mutated proteins, which might be caused by the SNP that is being incorporated in that particular case. Hence, with using the Sopma tool, you may compare the different levels of secondary structure between your normal protein and the mutated protein.